The purpose of this research paper was to find data that quantifies codon selection between synonymous codons, or in other words, how often one codon is chosen over others that code for the same amino acid. This data gives an idea of how genome-wide sequence evolution can occur, and it goes back to the concepts of evolution and natural selection that we learned about in class. The theory of evolution as proposed by Darwin was based on the concept of natural selection. Though natural selection is not the only way organisms evolve, it is the only way for organisms to adapt. These adaptations can lead to an organism's increased fitness, which means they have an advantage in their ability to survive in their environment. As we learned, natural selection requires 4 things to be true. The trait must be heritable, the trait must have variation, and differential reproduction must occur. All of this background knowledge that we learned in class is needed to understand the ideas in this scientific paper. The findings of this research can be applied to the design of synthetic genes and gene expression in biotechnology. In order to determine the selective advantage of some codons over others, optimal codons in tough genes and Salmonella were exchanged for synonymous codons. The amino acids studied were leucine and proline. These amino acids were chosen to be studied based on 3 criteria. First, multiple synonymous codons exist to code for it. As the sample portion of the codon chart shows, multiple different codons can code for the same amino acid. Second, the amino acid must be frequently found in the Salmonella. And finally, the codon usage for this amino acid is highly biased, or in other words, one codon over all the other possible ones is often used to code for this amino acid. For both amino acids, synonymous tough alleles were designed as shown in Figure 1 and synthesized. Altered tough alleles were constructed, amplified by PCR, and inserted into chromosomes to replace the genes. Bacteria was grown, and the fitness of the different bacteria was measured through competition.
The experiment found that preferences for certain codons in Salmonella are directly related to the bacteria's fitness levels, particularly its translational speed. Figure 5 shows translational time of the optimal gene tough A is indicated by the filled circle. The synonymous codons are shown by the square and triangles to the right of tough A, meaning that necessary translational time increases if these codons are present. The average selective disadvantage of synonymous codons in tough genes was compared to the number of synonymous substitutions using an R-squared value. An R-squared value compares what percentage of the variability in selective disadvantage can be predicted by synonymous substitutions. Figure 2 highlights 3 synonymous leucine codons and the selective disadvantage of a generation of Salmonella experiences with the presence of leucine instead of tough A. An R-squared value of 95% for all 3 regression plots suggests a strong correlation between synonymous codons and selective disadvantage to Salmonella's overall fitness.
Overall, changing the codons reduced fitness in the Salmonella by 0.01%. Although this change might seem insignificant, a small change, such as this one, can pick one codon as weaker or unfit. This is called codon bias, and is very prevalent in species of bacteria and yeast. This occurrence is not beneficial to us because in these species of bacteria and yeast, they cause infections to humans. So more in-depth research would help humans lessen these infections.